I think we can start now, I guess.
Okay.
Okay, ladies and gentlemen, good morning. I'm Lichen, the editor-in-chief of Myco-Asia Journal of Modern Mycology. Welcome to the Myco-Asia Masterclass. Today we have with us Dr. Sanjeeva Nayaka, Chief Scientist at CSIR-NBRI Lucknow in India. He's an expert of lichenology. Welcome to the program, Dr. Nayaka.
Thank you. Thank you.
And thank you very much for agreeing to give this talk. And we are still getting people into the platform. By the time we actually start with your presentation, there will be some 50 people. Before I hand over the platform to you, let me briefly introduce you to today's audience. Many of you are aware of your work. Many of you may personally know you, but for the sake of formality, I'm going to introduce you to today's audience. I'm going to read out from the prepared note. Dr. Sanjeeva Nayaka is a well-known lichen taxonomist of India. He has got a vast experience of over 25 years. He had initiated his career as a project assistant at CSIR-NBRI. Then he joined as a scientist there. Currently, he is the chief scientist and head of the PDSH division. He had extensively explored different phytogeographic regions of India, published number of floristic accounts, revised a dozen of taxa, and discovered 50 new species of lichens and reported over 100 species as new to India. He had also carried out biomonitoring, biodeterioration, bioprospecting studies on lichens. Dr. Nayaka's credibility can be assessed from his publication in reputed journals, around 224 publications. He has guided 6 students for their PhD, and he has handled 13 externally funded projects as a project leader. Dr. Sanjeeva Nayaka contributed immensely to the growth of lichenology, which is otherwise a neglected group in India. He established the Indian Lichenological Society as a founding secretary. He spread lichenology all over the country by training a large number of research scholars and faculties. I mean, more than 70 people have got trained under him in lichen taxonomy. He has organized national and international conferences, workshops and trainings. In addition to, he has delivered over 60 talks and published popular articles in regional languages. I listened to him when I was in Calicut, actually, for the first time, and recently Dr. Rajesh Kumar met him at KFRI, Pichi, Kerala. Thank you very much, Dr. Sanjeeva Nayaka. With this brief introduction, I hand over the platform to you.
Very good morning to you all. And first of all, thanks to Dr. Belle Damodar Chennai and other office bearers of Myco-Asia for giving me this opportunity. And I see among the audience there are several lichenologists, already established lichenologists have joined in this group, and I hope this talk will be helpful to all, both the beginners as well as the laymen, and also to the learned lichenologists who have joined in this event today. So let me share my screen. Can you see my screen?
Yes, sir. Yes, sir.
Hold on.
Yeah.
I myself unable to see the screen. Okay. Can you see it the full screen?
Yes, I can. Yes.
Yeah. So in my presentation today, the first half will be very general, very basic regarding the introduction of lichen, regarding their usefulness and then diversity and all. So the other half would be of much of the whatever so far India has done in the lichen research, and the what is left to be done. So this is good for both lichen researcher as well as the other people who are interested to take up the lichen research as a career, because in India we have done a lot of work, but still many things to be done. Uh, uh, Coming to the basics of lichens, the lichens are called in different names in different languages. In English itself, it is called as lichen or lichen. So it's a Greek word, and both are acceptable actually. But more often we use lichen as the name for the lichens. Okay, instead of lichen, we call lichen. Lichen is better. So in even if you see if you from different places, you can search the name in your language, but it overall means that stone flower. So in if you take in Hindi, it is called Patarka pool, or in Kannada Kallu Huvu, or Marathi Dagad pool. So the Dagad or the Kallu or the Pattar, they all mean stone. Okay, since these lichens lichens grow luxuriantly on the rock, they are probably called as the rock flower, the stone flower. But it doesn't mean that these lichens grow only on a stone or the rock. They also grow on various other substrates. For example, they grow on tree bark, grow on soil, sometimes they grow on leaf. Um, like that, they grow on many substrates. But to give a name to the lichen, it is commonly called as stone flowers. So, so what are lichens? Lichens, you all by this time know that is they are the symbiotic association between fungus and an algae. So algae sometimes we call as the both green algae, blue-green algae, or the cyanobacteria. Also we call as the algae, but it is a basically association between a fungus and a photobiont actually. The fungus we call as mycobiont, because myco is for refers to fungus. So mycobiont, whereas the photosynthetic partner that is present in the lichen, they are called the photobiont, which can be algae or the cyanobacteria. So these are the true primary symbionts, and there may be a other symbiont or the other photosynthetic organism as a third symbiont, that is cyanobacteria, that is called as parasymbiont, that will tell you in later. So whatever you see from outside, when you see a lichen thallus, actually you are seeing lichen. You are seeing actually fungal part. So algae is there actually inside. When you cut a section and see, then you will see the algal part or the photosynthetic partner. So the when these microbiont and the photobiont they come together, they change their appearance. They completely change. They undergo modification, and they start acting as a single organism. So they lose their original identity. This is the beauty of lichens. So then only we call it as a lichens, and this process is called as a lichenization. Okay. So if you see here in this picture, when you cut a section of the lichens, you will see like this. So the round ball kind of structure, what you see green, that is the algal cell, and the rope kind of structures, what you see here, that is the fungal hyphae. So you can again see here the upper portion only. Here in this portion only algal cell is restricted, only probably 10% of the whole volume of the lichen thallus, and 90% is made up of fungus. And fungus is the one which gives shape, structure, and um, color to the lichen mostly. That is why, and fungus is a dominating organism here. One thing we should understand here: the lichens are nothing but fungus, because the fungus is a heterotrophic organism. It has to depend on the other organism for the nutrition, either as a parasite or as a saprophyte, which grows on the dead organic matter. So here in this case of lichen, it is a symbiotic relation between an algae. So it's a kind of mutualism, or you can say the controlled parasitism. So that is why here the fungus is a dominating organism. That is why we classify the lichens under the fungal kingdom. 1/5 of the fungal species are lichenized for that matter. So whether this is algae and fungal association is a very unique association found only in lichens? No. if you see in the nature, um, there are other fungal and algal association is there. This is called as mycophycobiosis. For example, if you take an example of geosiphon in the first picture here, the extended finger-like structure, what you see, this is a fungus, and there inside, if you cut a section and see inside, the cyanobacteria is living inside this fungal cell or the fungal tissue. Whereas there is one more relation is there. It is called blockbacteria. So where we here the fungus is living inside the algal cells. So now both are possible. Algae can live inside the fungus, and fungus can live inside the algal cells. So this is the relation, one organism living inside the other organism. Whereas in case of lichens, this is the symbiotic association, but both are situated outside. You can clearly see here, algae is outside the fungal tissue or the fungal mycelia, whereas the fungus is also outside the algal cells. So of course, to draw the nutrition and all, say fungal mycelia make a haustoria kind of structure that penetrates into the algal cells and draws the nutrition, or simply sometimes they are attached together, they're close in contact, but they're not living one inside the one another. So this is the difference between the symbiosis or the symbiosis in lichen and in the other cases of the algal and fungal association. So lichens tissues are very delicate. They are the very poor fossil former. The very good fossils do not form. So that is why, even though before 415 million years ago, there was some fossil kind of structure for there, we could not clearly identify them. The researcher could not clearly identify them. So that is why the only after getting a very clear this association, clear fossil where the people could recognize the fungus and algae, then only we dated such a fossil to 400 million years back. So now we know that lichens evolved or the origin at least 400 million years, 415 million years back. Then they're there in the nature, they were there, and people are using it. People are, looking at it since a long time, but only in 300 BC, Theophrastus, he is also called as the father of botany, he termed them, he coined the term lichens to recognize them. So lichen, he called them lichens. So for the superficial growth of fungus kind of growth, epiphytic growth on the olive tree, but he did not clearly define what exactly lichens or how they should look. But this later on, the Greek authors, Greek literature people, they popularized the word lichen. They, in that process, they included all the other organisms. They also included moss, liverworts, fungi, seaweeds, corals, and all under the lichen. So gradually thereafter, before Carl Linnaeus also, people were studying the lichens. For example, Joseph de Tunford, he classified the group the lichens. Mostly he included lichens only, but also in between he included few bryophytes also. But John Jacob Delineus, he is the one who recognized lichens as a pure group of organism. All the lichens he put together, uh, excluding the bryophytes and other things. That is why sometimes people call Jacob Delineus should have been the father of lichenology. You can see the date. His date is before the Carl Linnaeus period. So then came Carl Linnaeus. You all know he's the father of modern taxonomy. He written several books, especially Species Plantarum and all. He recognized the lichens, and he recognized 80 species of lichens and put them under the 24th class of Cryptogames. Then his student Eric Acarius, he's called as a father of lichenology. So by the time microscopes were already discovered, Carl Linnaeus, he did not use much of the microscopes, but Eric Akerius used mostly the microscopes, and he identified several lichens, and he coined new terminology. He classified around 3,300 species of lichens into different groups. So, that is how he is known as the father of lichenology. But till the Carl Linnaeus period or the Eric Akerius period, they didn't know that lichens are actually made up of two organisms. Only in the year 1867, Simon Swedner, a Swiss botanist, he could demonstrate that lichens are made up of two organisms. Simultaneously, Anthony D. Berry also found that lichens were made up of two organisms. These two people are working together but in different places. Uh, um, D. Berry was the strong opinion that lichens were made up of two organisms. But Simon Swidner was the one who could encash this idea, and he presented in a conference. So, of course, this idea of dual nature of lichens was criticized in the beginning. It took long time to accept. It took almost 100 years to accept that lichens are made up of two organisms. So after discovery of two organisms, in the recent times thereafter, several other organisms which are growing in close association with the lichens are also discovered. For example, in 2016, there was a um, paper by Spreebill, so where he said that the yeast are the third partner very closely associated with the lichens, and he proposed the theory that lichens are actually made up of three organisms, not two organisms. Again, that was subjected to criticism. There were several papers later on, so which proved that no, these yeast, whatever he has discovered, is an actually parasymbiont. They are the usually lichens present. Lichens has several other organisms also. So like that, now we know that lichen is not an association of simply two organisms, but they are the association of several other microorganisms. It can be the algae. One already there is one more of algae or photoband. There can be an additional algae may be present on the lichen thallus surface. There may be bacteria. There may be yeast. There may be other fungus which are living in association with the lung with the lichen, and protist, virus, several other things. So now this association is more sometimes obligatory association. It is not that simply faculty association. And every organism which is associated with the lichen, they have a role to play. For example, sometimes we used to think how these lichens take up this nitrogen, because nitrogen is very important micronutrients required for the growth of any organisms. So now we know that lichens have the nitrogen fixing bacteria growing on their thallus. So like that, every organism which is associated with the lichens have a role to play. That is why the definition of lichens now has changed. We call now lichens as a micro ecosystem or the ecosystem. So in the 2020 Hawksworth and group, they came out with the definition of the lichens. Like a lichen is a self-sustaining ecosystem formed by the interaction of an ex-habitant fungus and an extracellular arrangement of one or more photosynthetic partner and an indeterminate number of other microscope organisms. So what we have to understand here is that lichen has two primary symbionts. One is that fungal part, that is mycobiont, and one photobiont. These two are important. They are primary symbiont. And you will get n number of other organisms association with the lichen. They are all parasymbionts. So if a fungus is growing on the lichen thallus, they are called as lichenicolous fungi. This is parasymbiont what I'm talking. And if a parasymbiont or the additional fungus growing inside the lichen thallus, then it is called endolichenic fungi. So like that, we know there are several organisms are already associated with the lichens. So the association of these two organisms, basically primary symbionts, algae and a photosynthetic organism, fungus and a photosynthetic organism, gives unique ability to lichens. So they can exploit a variety of substratum. They can grow on rock, moraine, concrete, timber, old building material, rusted iron, many other man-made substratum, even the plastic and all. So they can grow on um, harsh and hostile environment. They can grow on the hot desert as well as they can grow in the cold deserts like Antarctica or Leh Ladakh. So, but they're slow growing. Their metabolic rate is very slow, and that is because they are poor competitor. So they cannot compete with the other fast growing lichens. That they are the they look for a place which has a poor competition or the poor growth of other organisms. So they are poikilohydrous because they cannot maintain their water balance. If the harsh climatic conditions appears, harsh weather condition, they lose their water and they desiccate completely, and then they remain inactive. Whenever the water is available, they come back to life. That is why they are also called as resurrective plants. So as I told you, they remain physiologically inactive during harsh conditions. They can tolerate extreme freezing condition. They can grow even in minus 196 degrees centigrade. They can photosynthesize in the low temperature as well as in the low light also. This has been seen in Antarctica. In Antarctica are the lichens which grow in the cold desert areas. There the frost injuries or the cold injuries are not seen in lichens because they have the carbohydrates called as polyols, which protect these lichens from the freezing stress or the injuries. So because lichens are sturdy organisms, so they can tolerate extreme climatic conditions, lichens are sent to a space in a space experiments. So the lichen fungus experiment is the name of the experiment that lichens are sent to International Space Station. So this lichen, Xanthoria elegans, this is the picture what you see here. So this lichen was there for 1.5 year, and when they brought back to the land again, so they checked the how many are surviving. 71% of the lichens were remained viable. So, so then I'll quickly go through the uses of the lichens, their service to the ecosystem and all. One of the best well-known service is that that they are used in the soil formation. So that is again called as ecological succession. You might have read that during your college days. So the lichens are the pioneer organism which grows on the barren rocks. So once they grow and start growing on the barren rock, they produce some chemicals, or by physical process they cause damage to the rock surfaces. So these rock surface get deteriorated, and they break into small particles, and that is how this process takes place, this pedogenesis, whether it is a physical process or chemical process. So it makes a place suitable for the growth of the other organisms. And the next organism is to come is the bryophytes or the mosses. And then it makes a place suitable for the herbs to grow, maybe grasses and weeds. Then shrubs will come. Then shrubs are replaced by the trees. Like that, in a barren rocky area, will finally get converted into a climax forest. Of course, this is a very, very slow process. You may not visualize in your lifetime, but if you consider the geological scale, it is one of the important process happening in the nature. Then lichens serve as a food to several microorganisms or the smaller invertebrates, maybe snails and insects and all. So even it is a staple food for the deers, the reindeers in the temperate areas. You can see the carpet of lichen growing. It is called cladia. So especially in the winter when there is no grasses, the lichens are hidden, or the lichens are submerged in the snow, and these deers have a special smell and the sense to identify these lichens, and they dig out the snow and eat them, eat the lichens. Sometimes lichens are collected from the forest, and they are fed to the animals in the farmhouses. And the lichens also provide shelter to the several of the microorganisms. And some of the lichens which have cyanobacteria as a photobind, they fix nitrogen, and when such lichens fall into the soil, they form the increases the fertility of the soil. And then the lichens give a very good background for the camouflage. For example, you can see the a pepper moth is sitting over the lichen here in the first picture, and it is nicely camouflaged. Like that, this is a catidid insect. It is nicely mimicking the Asnia lichen here. So these are the small contribution of the lichens in the nature. And lichens are used for building the nest. Either, for example, you can see here, this is the hummingbird. Although nest is a very small nest here, and it has used maximum lichens for the building the nest. Whereas in this picture, you can see lichens are used for decorating the nest. So you people may be eating lichens, but you may not be knowing it, because the lichens are the household item in India. So it is used as spice. Either it is used as a khada masala or the ram masala. So, or it is ground into powder and used in the masalas. So if you carefully see some of the garam masalas and all, or meat masala, you turn the packet around and see stone flower. They might have mentioned as a stone flower for the lichen. So lichens is one of the important ingredients in the spice. It may be in uh, Lucknow kebabs, or in Chettinad masala, or Goda masala of this one Maharashtra. So lichens are used as a spice. So lichen as such don't give the smell. Don't be under the impression that if you take a lichen and smell it, it gives a smell like curry leaves or the coriander leaves. No, it's not like that. So when you take a piece of lichen and fry it in the ghee or in the oil, then it releases the aroma. So then it changes completely the taste of the food or the flavor of the food. So sometimes the people may be eating biryani or some nice dish outside, non-veg dish outside, and they try to mimic it at your home, but it doesn't give you same taste. That is because you may be missing an important ingredient such as lichen. So sometimes the lichens are the chef's secrets in the restaurants. And lichens also used in the perfume industry. Again, as I told you, it don't give a smell as such, but it's a process actually here. What they do, they boil the lichens in a big container. Here, this picture you can see, this is a picture from a city called as Kannauj here in Uttar Pradesh. It is also called as a city of perfume. So in the big container, you can see here, they put lichens along with the other herbs. Sometimes they purely use lichens and they boil it. So when that water vapor comes through this pipe here, you can see the pipe here, in between they keep an essential oil. This essential oil may be sandalwood oil or the rose oil. So the vapors mixes with that oil and gives a different smell. Now you can say that already sandalwood oil or the rose oil already has a smell, and why should again how the special how what is the specialty of lichen? What is the smell of the lichens now? So lichens has a kind of smell. Only it gives better smell when mixes with the sandalwood oil or any other essential oil kept in between here, and it's a completely different smell, and then it gets sold for the very expensive price. So this is how the people use in the perfume industry. Lichens also used in the dyes and all. For example, litmus paper, what you have used in the laboratory, acid-base indicator, it is prepared from the lichens. The lichens called as Rossella montagni or the Rossella other species. Switzerland is still a major country to produce the large amount of litmus paper. Of course, in the nowadays we have the pH meter is there, or the synthetic pH papers are there, but earlier the lichens dye was used for the preparation of the litmus paper. And even the lichens are used for the dyeing these silk and the wool. So it is actually a process where they boil the lichens with the texture. So this is based on the type of process. Sometimes you can simply boil with the plain water, or you can also treat with the ammonia. Sometimes in the villages where there is ammonia is not available, they use the cow urine. So this is the type of the process what you are having, and the duration of the process. Based on that, the lichens give, and also depending on the different species, it gives different color. And lichens are used in the aesthetics. Um, so in the sometimes the tribal people in the some ceremonies they wear the lichen on their head and the body. So if you're living in a temperate place, if the lichens are plenty around you, you can bring such lichens and put it in your garden for the beautification of your garden also. Apart from that, you can make nice artifacts using the lichens. Even the room freshener or the potpourris you can make using the lichens. So lichens are also traditionally used as a medicine. And if you take any literature throughout the world, there is a mention of the lichens for the medicinal purposes. So even in India, the Nigantus or the Ayurvedic literature, even Vedas had a reference of lichens for being used as the medicine. In Sanskrit is called uh, Shailya. So in the old literature, you can get the reference of lichens. And it is also important ingredients in the Chinese medicine and also Unani and homeopathy. And this is Acharya Balakrishna. He has written a very nice article in Hindi. You can be easily accessible it is. So he has said that lichen can be used for the gray hair problem, headache, eye diseases, urinary diseases, and many things he has mentioned. Apart from that, lichens are also used by the tribal people for the wound healing or for the stopping the bleeding. So coming to the diversity of lichens, different people at different time give the different estimations, but you can say that in the world there are 20,000 species of lichens are present, and tropical forests are actually the hotspots for the lichen diversity. Sometimes you can get about 600 species of lichens per hectare. And then up more, about 50% of these tropical areas, tropical forests are unexplored for the lichens, because these tropical areas are most inhospitable or more inaccessible places. That is why they are underexplored for the lichens. Coming to India, so at present we have 3,066 species of lichens, 4 subspecies, 28 varieties, 1 pharma, and totally we have 3,900 3,099 taxa we have, which makes if you 15% of the world known lichens. It is distributed under 490 genera and 93 families. About 550 are endemic to India. You don't get them anywhere else. So in India, Western Himalayas, northeast part of India, including Himalayas and northeastern states, Andaman Nicobar Islands, and Western Ghats are considered as the hotspots for the lichen diversity. And in India, you can see that the exploration of the lichens or the discovery of lichens, addition of new records and all. So you can see the known lichens from India are increasing year by year, and this growth is exponential. So that is all because that we are discovering several new species or reporting new records every now and then. And these are some of the important people from India who have contributed to the growth of the lichen research. It all started from K. Biswas. This is just after the independence or just before the independence period. K. Biswas, who was actually the superintendent at Botanical Garden at Howrah. So he, with the help of Dr. Avasti, prepared a first list of lichens for India. Dr. Avasti is actually went to K. Biswas for learning about lichens. And then Dr. Avasti started independently after taking his PhD from the Colorado University. Dr. Avasti established the Lichen Research Center at Lucknow University. And for a long time, he was the pioneer. And that is why he is called as father of Indian lichenology. So after some time, PG Patwardhan at Agarkar Research Institute started lichen research. He took the help from the Manson E. Hale from the America. Then KP Singh is a student of Avasti only. He carried out the lichen research at Botanical Survey of India. He was followed by GP Sinha. Now at Botanical Survey of India, Jagdish Ram is working. And then at NBRI, Dr. Ajay Singh started working on lichens. Um, and then he was succeeded by Dr. Upreti. Now at present at NBR, I am looking after the lichen research. Of course, there are many people who are working in different places. They are well established, skilled lichenologists, thorough lichenologists are there in India they're working at, for example, Dr. Yogesh at Jaipur, Dr. Siljo at KFRI, and Damari at Bodoland University, even Biju Haridas at Kerala, Vinayak at Karnataka. Like that, we have several people working on lichens in different parts of the country, and you can say around 200 plus people are working, and several of them are who are beginners. They're taking the help from the NBRI for the identification of lichens or the other aspects. So coming to the prospects of lichenology, so the what are the possibility or the scope for the lichen research in India? So it can be divided in two main groups: basic research, um, basic research where where the taxonomic work can be done under this basic research. The basic research are mainly for the understanding or the generating knowledge, whereas the applied research, which has applied component, which is applied in the sense useful component for the society or the environment. So they are the applied research. It may be biomonitoring studies, air pollution, or the climate change studies, or it may be biodeterioration or the bioprospecting. So I will go one by one. For example, like in systematics, or the it may be it is a taxonomy, floristic studies, or the revisionary studies. Floristic studies means documenting the all plants living in an area, or in case of lichens, a floristic study means the documenting the lichens growing in an area. So there, that is a floristic study. It can be a simple list or inventory of the enumeration, or it may be elaborate flora, which includes illustrations, identification keys, ecology, and many other things. So in case of lichens, we call lichen biota or the mycota. Since we don't consider lichen as the plants, we don't call them flora, but sometimes there is a controversy again. Some people again call as a flora. So see, if you see this graph here, you can see that Uttarakhand has the maximum diversity of lichens in India, whereas the right side of the graph, there are no lichens, or very few species of lichens are there. So it doesn't mean that in this area there are no lichens. There are lichens, but nobody has studied that. So since already very less people are working on lichens, so people have not concentrated or they not studied the lichens of these area. But if somebody take up the studies in this area, we can come out with the lichens in these areas also. So this is simply one of the example for the floristic work. So and also scope for the work. I'll give you a few more examples, and these are some of the places where the floristic studies have been considerably well carried out. I'll not read the names. And for example, I started my career with the Himachal Pradesh, studying the lichens of Himachal Pradesh as a research scholar. So when we began in the year 2000, there are only 309 species of lichens were known. When we completed the studies after 5 years, or even after that, we are discovering few species, new species and all. Now we know that 429 species of lichens are present in the Himachal Pradesh. We can see that around 120 species are added to the list of lichens of Himachal Pradesh. This is the scope of floristic work. Like the Eastern Ghats, although it is a very interesting area with several endemic species and all, there are no records of lichens, or very few records of lichens. When we started our work with the help of Dr. Madhusudana Reddy from Yogi Vemana University, we could list only 31 species of lichens. And by 2016, we know that there are 225 species of lichens are there in the Andhra Pradesh. So we again reported several new species and the new records from the Andhra Pradesh. So like that, Uttar Pradesh was, although since it is a plain area and the harsh climatic condition is there here, so people that the lichens are not do not grow in Uttar Pradesh. So that is why in 2010, according to Singh and Sinha, there were 41 species of lichens were there. Then we started working on the lichens, and then in 2014, I came up with the book which had 135 species, but we are still updating the lichen list, and at present we know that 185 species of lichens are there in Uttar Pradesh. So in Gujarat, earlier only 2 species of lichens were known. Now we have about 95 species of lichens are known. So this is the scope. Goa again, when we started, there are only 48 species of lichens were known, and now at least we have around 160 species of lichens are known from the Goa. So this is again Mizoram. You can see 2010 2 species were there. Now we have 185 species. Tripura, there were 0 species. Now we have 29 species. So what I mean to say here is that there is a lots of scope for simple study, that is floristic study, in India. In India. So some of the places like special vegetations like Kudremukh, the Kudremukh in Karnataka, you see this kind of vegetation. These are called shola vegetation. They are very interesting area, interesting niche for the lichens, but there are no reports of lichens from these areas, and this is a potential area for the exploration of lichens. Apart from this, it is also necessary to revisit some of the places which have already been studied for the lichens. For example, Nilgiri Biosphere Reserve. The most of the work that has been carried out in this area is very old, maybe during the time of Dr. Avasti. Now there is a need for the revisiting this place because lot of tourism activities, urbanization might have affected the environment of that area. Several lichens might have vanished from that area. That is why, in the current scenario, there is a need for revisiting such areas which have not revisited for a long time. So these are some of the high altitude peaks in Western Ghats, just for an example, which needs exploration for discovery of interesting or the novel species. And coming to the revisionary studies, what is revisionary studies? Revisionary studies is a pure taxonomic studies. So that is a restudying in detail the already studied taxa. That is for the updating the taxonomic and the related information. So what happens? Taxonomy is a very dynamic and evolving field. So whatever our understanding regarding a species or the taxa or the genus, it may change after some time. So, for example, when Dr. When Carl Linnaeus or the Eric Cacarias or these people, whenever they're studying the lichens, they used only hand lens or the primitive microscope. But now we have very advanced microscope. We have, apart from the light microscope or the stereo zoom microscope, we have scanning electron microscope, confocal microscope, several things. The structures which we could not visualize earlier, now we can visualize them better. So apart from that, people were using earlier the taxonomy, classical taxonomy or the microscope based taxonomy. Now we have tools like we have chemotaxonomy, we have molecular systematics, molecular taxonomy. So our our tools, techniques, skills, and in studying an organism has improved. So we are adding a new character to a taxa which has already been studied. That is why there is a chance that whatever our understanding regarding a taxa may change. So that is why updating this information is very much important. So while doing so, we will come across with the new species, sometimes new higher taxa, genus and families and all, or so sometimes we felt those two specimens are same specimen, same species, which may get separated into different species, or two species or more species which we thought different may merge into same species. So these are the things happens and keeps happening taxonomy. That is why the revisionary studies are very much important. So these are some of the taxa group or the families which are well studied in India. There are many. To put some of them, I have put it like that. And for example, Graphidaceae, this is one of the largest group of lichens in India, in the tropical countries. They are the crustose lichens, and these institute like ARI, Pune, NBRI, even BSI, these institutes are regularly, constantly working on this group of lichens, but still it is not completed, and there are several interesting groups yet to be studied. So, so there are again taxonomic controversies, or the split is there. Earlier, thyllotromatide lichens are under the Graphidaceae. Now they are, they are different. Now they are merged together. So, like that parmeliad lichens, my senior Dr. Divakar, who is at Spain now, he studied this group very well from India. So earlier it was under one genus. Now we know that these lichens are spread in the 25 genera, and we have now 209 species from India. So, like that, Pyrenocarpus lichens are the lichens which produce perithecia as a fruiting bodies. So, this group, although constantly or regularly people are working on this, still there is a lot of scope to work on this. 337 species we know now under 38 genera, but some of these genera are still to be explored. So folliculous lichens, again, very interesting group. The lichens which grow on the leaf, evergreen leaves. So, very interesting group. And these lichens are also indicator species. They indicate the health of the forest or the open canopies and all. So now we know that over 150 species of lichens are there. Again, the tropical areas, whether it is Northeast India or the Western Ghats, there's a lot of scope to work on this group of lichens. And as I already told you, there is a need for the revision of Graphidaceae, again, Physiaceae. In our institute, NBRI, we are already working on group Physiaceae, Roxalaceae. Dr. Silja Joseph, and he's already working on this group. Arthonials also, Hematoma also is working. And there are many other groups to be revised, and Pyrenocarpus, as I told you. And so, as I told you, rapid taxonomic changes is happening in lichens in the world. So that is why in India we have to match our taxonomic research with the world trend. And so again, one more important thing I want to tell you here is that we are still dependent on the classical taxonomy, and it's a high time now that we have to incorporate molecular systematics to the our study. And it can be started with the simple ITS sequences, and also including protein coding markers and other things. So DNA barcoding for the lichens can also be done, which is very easy because ITS is considered as a universal marker for the lichens. That can be done, and such studies are required. Uh, so, so the molecular study is important again because the lichens shows very less morphological characters. So morphological characters alone is not sufficient. That is why these molecular studies can be supplemented with our classical studies. And this group, Lichenicolous and endolichenic fungi group, is very important groups. As I already told you, Lichenicolous fungi are the fungi which grows on the lichens, whereas the endolichenic fungi, the fungi which grows inside the lichen thallus. So Lichenicolous fungi shows the symptoms of infection, whereas the endolichenic fungi will not show any kind of symptoms of its presence inside the lichens. These lichens, these fungi, they are known as known for the production of unique secondary metabolites. Already lichens produce several metabolites, and in addition, lichenicolous and endolichenic fungi also produce unique secondary metabolites, and these metabolites can be exploited for the bioprospecting. For example, some of the metabolites extracted from this group of fungi, they are known to be good anti-cancer, antiviral, antibacterial, antifungal, and anti-Alzheimer activities they have shown. So again, these group of fungi not only taxonomically important, but again they're important as in the perspectives of evolution, specificity, affinity, ecological preferences, biotic and abiotic factor, how they influence in the growth. They're very much important. So now the Lichenicolous fungi number which I mentioned, it's very less. We know now that around 200 species of Lichenicolous fungi are known from the country now. So, like that, endolichenic fungi, when the people work, they mostly concentrate towards the unique secondary metabolites produced by the lichen, endolichenic fungi. Taxonomy, they don't give much importance. Even then, we have around 180 endolichenic fungi discovered from the lichens from India, from throughout the world. And coming to the biomonitoring studies, it is air pollution and air climate change studies. So in air pollution, see, lichens are very much sensitive to the air pollution. It is important to understand why these lichens are very much sensitive, because as I told you, 90% it is made up of fungi, and when 10% is only algae. So this 10% algae is very much important and sensitive. So if algae dies, whole lichen thallus dies. So what happens because of the pollution, the chlorophyll content, it has the iron magnesium molecule in the inside. The chlorophyll, this magnesium molecule will get replaced by the it is other molecules. That is why the chlorophyll gets converted into pheophytin. Once it gets converted into pheophytin, the photosynthetic efficiency reduces, and that is how the lichen gradually by reducing the this algal cells dies, and then the whole lichen thallus dies. So again, the lichens do not have root kind of structure, although some of the folios lichen have these rhizines, root-like structure. They're used only for anchoring, attachment. So they don't draw the nutrition from the substratum. So that is why lichen has to depend on the atmosphere for its nutrition. Whatever the nutrients dissolved in the humidity or in the atmosphere, it is directly taken by the lichen thallus, especially during the wet condition. So lichens do not have these protective layer like cuticle or something like that. That is why the lichens are much exposed to the air pollution, and they directly absorb these pollutants into their thallus, and that affects the algal cells, and finally lichen die. And when they die, they bleach, they change their color, they bleach, they peel off from the substratum. Like that, it indicate the air pollution. And if you have the earlier record, you can compare the earlier record, earlier observation to the present observation. You can clearly see that lichens have changed, lichen diversity, the community structure have changed in that area. So so we have done our studies in the using the lichens. We have monitored the pollution in the different cities of the countries, but there are many more cities are there to be explored, and we have estimated the heavy metals or the pollutant accumulated in the lichens. Especially there are some lichens, few species which are pollution tolerant. We bring such lichens to the laboratory and do the metal estimation studies. And sometimes when there are no lichens in the area city center, during that time we bring the lichens from an unpolluted area or the less polluted area. They will fix them, we'll put them on the board like this, cardboard like this, and put it in the different areas, trophic areas and all. So then we leave it there for 1 month, then bring them to the laboratory and then do the heavy metal studies, and we can clearly see the pollutant accumulated in the lichens. With that, we correlate with the pollution in the different places of the places. Like that, we have studied the several places. Like this is the example of Lalbagh Garden. When we studied compared our study, the 18 years study, uh, so we could clearly see that the number of lichens have changed, and also the community structure were also changed. So custos lichen finally became very dominant in the present condition. So because custos lichens are much pollution tolerant. Like that in Kolkata Botanical Garden also we have seen that in 1867 there was a study by Nylander. When we studied in 2005, we could see the complete change. There were earlier 53 species. Now, in 2005, there are only 21 species. Again, the custos lichen finally dominated this area. So, and apart from the heavy metal and the simple lichen documentation studies, we can also make lichen mapping and all. The PAH, PAH is that polycyclic aromatic hydrocarbons. So these are again, lichens are capable of accumulating that. They are again dangerous molecules, pollutants for the both for the human health as well as for the lichens. So what is our scope for the study in the air pollution? There are many cities in India has to be monitored for the air pollution using the lichens, and there is a need for the calibrating the pollution load in the lichens as well in the environment. So what I mean to say that when you estimate a heavy metal or the pollution load in the lichen, you should be able to say what is the level of the pollution in the air. So that kind of calibration is needed. And there are several indexes like indices like IAP, LDV and all. So these are simple, but very systematically has to be done. This has to be incorporated into our studies and come out with the quantitative data. So apart from that, we have to use the modern tools like global information system or the satellite based studies. We have to um, incorporate, and we make a different models using the heavy metal or the pollutant accumulated with the lichens. So this is one of the example of IAP. I'll not go into this. And coming to the climate change, like any other organism respond to the climate change, lichens are also very much sensitive. So what we have seen in our earlier study is that as the climate change happens, there will be increase in the lichen preferring the warm climate will increase, and there is an increase in the epiphytic lichens, and especially one with the green alga containing one. Among the green alga, tentrified lichen containing lichens will increase in their number. So apart from that, it has seen that lichens will have these several the changes in their metabolites. For example, lichens which are going in the extreme conditions or where the climate change is really happening, there they will have the increased amount of the metabolites such as asnic acid, perlotolic acid, fumer potocitric acid, phenolics and all. Even they will have the UV filtering metabolites also they have. So this has been seen. So for we don't have in India much of the studies for the climate change. That is why we don't have even have the baseline data. So therefore, if somebody is interested to take up these climate change studies, there is a lot of scope here. Again, we have to generate the quantitative data. We have to study the chemistry in detail related to the climatic condition. Again, the community structures we can study, and there is an also need for establishing permanent plots for the monitoring the regular happening in those plots. For example, you can see this is the open top chambers. They where this is the hexagonal structure, glass structure, which is kept on the lichen, and it temporarily increases the temperature of that this inside here, and we can monitor the change happening on the lichen thallus inside here. So this is hexagonal open top chamber that has to be uh, so included in our study. So these are the NBRI has a collaboration project with SAC Ahmedabad. So where we have established permanent plots on the hilltops in the mountain areas, and these permanent plots are monitored by the satellites. The changes here happening on the lichens and the mosses here, they are monitored directly by the satellite here. And this is a project result yet to come, and this is one of the important studies that has been carried out. And coming to the biodeterioration studies, as I told you in the beginning, you just remember the succession example I have given, like can grow on the rock and they break the surface of the rock into soil particle. So similarly, the lichens which grow on the rock monuments, they cause the deterioration, and it is very much important to remove such rocks. Apart from that, it causing the damage to the rocky structures or the monuments, they also give a very odd look, shabby look to the monuments. It is very much important to remove them. So from India, very few work has been carried out on this aspect. There is a lot of studies for also possible in this, especially towards conserving our monuments. So and then the other studies like ecology and conservation, quantitative ecological studies are very less in India. So where we put quadrates and the transects and such kind of studies. So quantitative studies are very much required to identify which lichen is dominant, which are the very rare lichen, whether any lichen require a conservation attention or something like that. So to assess this, ecological study or the quantitative ecological studies are very much needed, but such studies are very much less in India. That is why we don't have the quantitative data for any of the lichens, and conservation is very difficult. So none of the lichens in India are listed in the red data book. You may be knowing that IUCN red data book, which includes the um, includes the species or the organism which are under the threat of existence. It may be endangered or extinct or something like that. So none of the Indian lichens are there in this data. That is because of the data deficiency. And there are because we never done a quantitative data, but now there are other methods are available for including the lichens under the IUCN category. So we have to adopt that. Bioprospecting, bioprospecting sky is the limit for that. And I have already told you that lichens are used in the traditional ethnic medicine. And based on that, the several lichens are also tried in the laboratory for the biological activities. Because lichens produce more than 1,000 secondary metabolites, many of them are unique to lichen, and many of these secondary metabolites are biologically active. So antimicrobial activities or the antioxidant studies are the most common studies that have been carried out on the lichens, and lichens have shown potential uh, regarding potential activities also. Apart from that, anti-inflammatory, antipyretic, anti-tyrosine, anti-analgesic, anti-proliferatic, even the lifestyle diseases may be this anti-diabetic, cardiovascular, liver protective. Several of these studies have been carried out, but at all at a primitive level, and there is a need for carrying forward such kind of studies. So we have very less product from the lichens, although lichens are very well known for their biological activities, but very we have in the market very less product. And sometimes back, and at MSSRF Chennai, and even at ARI, the product development has been tried, but no such product has been come out yet. So so as I told you, there is a lot of scope for the bioprospection studies. Um, Apart from that, what is very much important here in the bioprospection studies is that we have to come out with the novel molecules. If not novel molecules, we have to come out with the molecule that is responsible for the particular biological activities. What has happened so far, if you take up the literature and see, they simply use the crude extract and then they'll list the biological activities. But here we have to come out with the uh, uh, particular molecule which is responsible for the biological activities, or we have to come out with the fractions which is responsible for the biological activities. And also we have to study the mechanism of action, how it takes place. And apart from that, we have to also find out the genes that is responsible for the for the production of that particular metabolites. So as I told you, lichens are very slow growing, and the large amount of biomass is required for any biological activities, and production of this biomass is also very much important, either in the laboratory condition or in the nature it is not possible. Only in the biology, in the laboratory condition, we have to grow this biomass. So it can be done. See, in the lichens, these metabolites are produced by the alga, I mean the fungal part, and these fungal are very comparatively easier to grow in the laboratory, in the culture. So they're also fast growing. We have to grow this microbiome, and also we should to make them produce these particular secondary metabolites. That is very much important. Such research has to be carried out. As I told you, we have to find out the pathways and the enzymes responsible for the production of metabolites. And the other possibilities, other scope is that whole genome sequences are very less in India as well as from the world. So the whole genome sequence has a lot of scope to be scope. Whole genome sequence gives answer to many of your questions, whether it is evolution or whether it is a genes responsible for the production of metabolites. And the other aspects is that lichenometric studies, forestry, physiology, and lichens growing in the special habitats, maybe sacred grove or the chola vegetation. So these are the other scope for the studies. And other thing is that digitalization of our herbarium in India. We have in several places very good herbarium, like in ARI, we have it LWG, that is in NBRI Lucknow, in Botanical Survey of India. we have several lichen herbarium that all need to be digitalized and brought into the single platform, that is virtual herbarium, or making a consortia of herbarium. This is one of the scope for the future studies. So this is the virtual herbarium of North American lichen herbaria. Like that, India has to come out with the herbarium for India. So with this, thank you. There are lot many scope for lichen research to be carried out in India. This is some of the examples I could give you, and this is good for both the lichenologists of our country who are already into lichens, and also for the beginner who wish to take up lichen research, or if somebody get inspired by this talk, you're most welcome. You're welcome, and you can always contact me. Thank you very much. And if you have some questions, you can ask me.
Thank you, Dr. Sanjeeva Nayaka. I hope I am audible to you.
Yes, yes, you are audible. Yes.
And thank you for that authoritative, insightful and energetic presentation on lichens. We are grateful to you. Before I allow the audience to ask questions, can I ask you a couple of questions, please?
Yeah, please go ahead.
Yeah, maybe you can switch on your video, please. Thank you.
Hold on.
Yeah.
Yes.
Great. I got excited when you talked about your collaboration with the Space Application Center, Ahmedabad. Then you were, and in that slide during that presentation, you said there is a technology where you can detect lichens from space by GPS, right? Am I getting it right? What is the resolution? What kind of information we get from the space?
Um, See, those permanent plots are there at the hill tops in the summits of the mountains, and they are monitored by the satellites which are there at the space, and these lichens and the any other plants or the organism, they emit spectra. These spectra are read by these sensors which are there in the satellites, and they give the different signatures. Through this, they monitor the whatever happening there in that spots, and exact resolution and the exact kind of thing they can only tell, and I'll not be able to tell you much regarding that. But that is happening, and this is very successful, and it's a long-term process.
Okay, thank you for that. My second question is when Dr. Manjusha gave a presentation some months back from Dr. Manjusha from Kufos, she mentioned that there are some marine lichens. Could you give some information about, because I am from an oceanographic or a research institute, and what kind of research that might have been conducted so far about marine lichens?
See, marine lichens, there are two kind of now marine lichens. See, one is that lichens which grow around the seashore. Okay, so they're not purely marine. So sometimes in the seashores you have coconut trees and the other vegetations. The lichens which grow on those trees also considered as a marine lichen, or the lichens which grow in the mangrove vegetations, they are marine lichens. But purely the marine lichens are the one which grow on the rocks in the seashores, and which are highly influenced by the saline water. So some of the lichens called as Verrucaria, they grow in the rocks on the seashores, and there we don't have purely submerged lichens, but at least these lichens for partially for some period of day they submerge in the water, and sometimes they are exposed. So the first kind of studies is the taxonomy study only. So the where how many species are the what kind of species are growing in this marine environment. This is the first study. Second thing is that ecological studies only. See, to their resistance to the salinity, and how they probably withstand that. Not much studies has been carried out in the marine lichens.
Thank you for that, Dr. Sanjeeva Nayaka. Ladies and gentlemen, now we are lucky to have two giants of lichenology amongst us today. Dr. Udeni Jailal, the co-founder of MycoAsia, is with us in the audience. Professor Jailal, can you please give your comments and questions if you have?
Yeah, I am here.
Yeah.
I don't know. Am I?
You're
Hello.
audible, yeah. You're
Am I?
audible, yeah.
Okay. Yeah. Dr. Sanjeeva, nice to see you after a long time. And
Yes,
thank you
yes.
very much for your nice and very informative presentation. I have to ask one more general question. Actually, this is regarding funding opportunities. How about the Indian governments? You can apply for any kind of grants related to lichen research, and how about their funding possibilities?
Yeah, funding. We are also struggling with the funding, but it's very highly. It's highly competitive. It is not that easy. I'm in a research institute and I'm in a CSR lab. So for me, funding opportunity again reduces. For example, I have only two funding agencies. One is that DST. DST again into two. That is the Department of Science and Technology, and SCRB we have, and Department of Biotechnology we have. Only these two, three are the funding agency for me. And we have in-house funding agency. Again, if we have a good project, highly competitive project, then only it's positive, possible. Even SCRB is Science and Engineering Research Board. They are much liberal, but again, your project has to be sound and well written. Then they support the taxonomic research. Whereas the biotechnology, Department of Biotechnology, it's possible, but you have to have the biotechnology component in there. But if the people who are in the UGs, I mean this one, universities or the colleges, they will have one more addition. The UGC gives them the grant. I don't know if still they give or they reduce now, I mean stop now. UGC gives the grants. Um, and these are the very few opportunities we have. Can't help that issue is there. Sometimes the state biodiversity boards give the projects funds that also people can avail. So yeah, and we have, but it is not too high or the very too not very less. Also, we have fairly. Yeah.
Thank you, Dr. Sanjay, because even in Sri Lanka also it's nowadays very difficult to apply for the grant and always looking for applied research, but not for the basic researchers. So that's why I asked that questions related to this funding opportunities.
Yeah, even for us also, we have to have the applied aspects or the things should be there. But as I told you, even in the basic research, if we have really interesting aspects, then it's possible. As I told you, SERB, they are good. They fund these basic research, taxonomic research. They fund. That is one good thing about that organization.
Okay, thank you, Dr. Sanjeev, and congratulate all the Indians who won yesterday match as well. Thank you.
Yes, yes. Yeah,
Okay.
congratulations to all Indians for winning cricket T20.
Yeah,
Yeah,
thank you,
yeah. Okay.
thank you, Jaila, Professor Jaila. Thank you
Okay.
very much.
And
Nice
thank you,
to,
Shenoy, for giving this opportunity. Okay.
yeah,
Thank
nice
you.
to hear you. Now we'll hope to see you soon. Uh, Dr. Sanjeeva Nayaka, you mentioned about some funding agency. How about MOEFCC and MOES?
MOEF, that used to be a good funding agency. Now they're not giving. They give only special projects, innovated projects with a special scheme and all. Otherwise, regular projects they're not giving. So that is one thing. MOES, that is Earth Science can give some projects, but you have to orient your projects towards their objectives, their mandate.
Thank you for that. Dr. Srivastava, please ask your question.
Oh, okay. Good morning, sir. Dr. Damodar, thank you for making such a nice Sunday. Every Sunday is so educative, so informative. And we can see so many people after the Indian T20 win up there to watching this. My point is just for the youngsters that such important is lichenology field in India. There is so much of scope. If you see the continent of Europe, which is very, very small, the whole continent has been mapped for the lichens, and they can monitor the pollutions starting from UK. The whole Great Britain has been mapped. So in India also that prospect does exist, though we are a big country. So as Dr. Sanjeeva has beautifully shown, the biodiversity that exists, and we can take small regions and map the entire country and see the pollution and anthropogenic activities, industrial activities. Very nice work, Dr. Sanjeeva. I must congratulate you because this is a very difficult task. As far as the impetus to this lichenology research, I feel that for the youngsters particularly, you have to incorporate three main techniques that will give momentum to this field. One is confocal microscopy, because you can really use the dyes, the antigenic dyes, which would show up, glow up the mycological partner, which is the most important thing if you are doing for the bioprospecting or for otherwise also. The other is bioinformatics. So, and bioinformatics would lead to a lot of PKS studies, and we can identify the main uh, genes that are responsible. And the other thing is MS, because by mass spectrometry, you can give a huge amount of data and you can get an idea as to what is the metabolite that is specific to this lichen. So these are some of the points that I thought I can make and add to this. So it was an excellent lecture, Dr. Sanjeeva. I learned a lot. And again, thanks to Dr. Damodar for making such a wonderful Sunday every time. Thank you.
Thank you very much. Nicely said. And certainly these aspects, techniques can be included, confocal, bioinformatics. Bioinformatics is very much needed nowadays. So without that, it's difficult to, present your data nicely. So yeah, thank you very much.
Thank you, Dr. Srivastava. You always come up with some more nice questions and insightful comments to actually enhance the quality of discussion we have at MycoAsia. Thank you, Dr. Srivastava. Dr. Gunjan Sharma, you may please ask your question now.
Thank you. First of all, thank you, Dr. Nayaka, for a wonderful presentation, very informative. I have a couple of basic questions. So should I ask all of them together or one by one is fine?
It one by one is fine because I forget. If you
Okay.
ask a series, okay.
Okay, sir. So first of all, you mentioned that so far there are very like either zero reports or very limited reports from North, like India, like Chandigarh, New Delhi. So do you think the polluted environment or the harsh climate or the heat of Delhi has any role in it? Because lichens prefer moist and humid environments. Is there some connection?
Yes, certainly pollution and the climate has a role to play in the diversity of lichens. But what we have seen is that even in such a climatic conditions, there will be lichens growing. They grow especially in the old monuments and all. In Delhi, especially, you have several monuments. So what happens if you see carefully these monuments? There are lichens growing. So they're the lichens which have the cyanobacteria in them. So although the climatic condition of the Delhi is harsh, you don't get the regular lichens. You don't get luxuriant growth of the lichens, but there are some lichens which are of special type. You have to hunt for them, especially the lichens, especially these monuments, for example Hazkas and all. If you go, so I have seen myself, if you see the monuments brick carefully, see sometimes you see the brownish blackish patches on the bricks there. You tend to ignore them as the algae or some dark spots or something like that. So if you carefully see them, they are the lichen. They are the lichens belonging to genus maybe Peltula or Endocarpon or something like that. So they have the blue-green algae in that. Such kind of lichens you will get. And if you try to go to some vegetation areas, so there is a possibility of getting the lichens like such as Pyxine, Brunneria, which are very tolerant, pollution tolerant, as well as the climate tolerant lichens will be there. You can find them. So you don't get luxuriant growth of lichens, but you will get few lichens.
Okay. The next question is again very basic. So these endolychenic fungi which you mentioned,
Yeah.
how different they are from the dominant microbiont in a lichen?
Yeah,
Like,
see,
it could be the same species or they have to be different?
no, they are different. They are different only. That is why we call them endolychenic fungi. See,
Okay.
we can't see them through the microscopic studies or through the through naked eye. So that they come only during the culture. What we do there? See, like you have endophytic fungi, like
Yeah.
you have endolychenic fungi. So when you take a piece of lichens, you nicely sterilize, do the surface sterilize and everything. When you put them on the culture media, then you get different growth of the fungi around that fragment of lichen thallus. So there you will identify those fungi. So certainly they are different from the main fungi that is present in the lichen thallus. So uh,
So how, like we have demonstrated reports on the role of endophytic fungi in plant growth. So do endolychenic fungi also have some reports or some articles which I can refer for plant growth?
So very less. I don't think you will get role of endolychenic fungi in the growth of lichens. Such reports are not there. I don't think it is there. But the endolychenic fungi, the different metabolites they produce, the diversity, those things, their biological activities, those literature, there are plenty. Not plenty. There are many. You will get it. But the role of endolychenic fungi in growth and the defense of the lichens is very less or almost nil.
Okay. Thank you, sir.
Thank you, Dr. Gunjan. Pinaki Chattopadhyay, please.
Yes, Pinaki, tell me. You ask your question.
Okay, Biplab Mahato, please.
Yes, sir. Sir, good morning, sir.
Yeah, good morning.
Good morning, sir. Sir, actually, sir, my question was that in the high altitude areas, we generally found fruticose lichen abundantly, but in the plains and some in the lower elevations, we doesn't find fruticose lichens, whereas we found the abundance of crustose and foliose lichen. Is it something? Why is it so?
See, if you say abundance, it's a qualitative term. I'll tell you why. So if you see the lichen diversity of any place, so crustose lichens are dominating. Then comes the foliose, then the fruticose. For any time, anywhere you take, fruticose lichens are very less. Now, in the lower altitudes, you will get only crustose and the foliose lichens. But as the altitude increases, you will start getting the fruticose lichens. If you see the proportion, again, fruticose lichens are less, but only thing is that the fruticose lichens start appearing at the higher altitude. Um, so because they are very much sensitive to the microclimatic conditions where they grow, they need much low temperature. They need much of the humidity and shade conditions. So these conditions are very much prevalent in the as the altitude increases. But if you take the lower altitude places, there the temperature is more and the harsh climatic condition is also there. And that is why in the higher altitude, these conditions are more prevailing, that humidity, temperature, light conditions and all. That is why they are more visible, though prominent there in the higher altitude places.
Okay, sir. Thank you, sir. Or sir, one more question, sir. That is in case of sir DNA extraction for lichen, we can do it from the thallus, or we need to culture the fungi particularly or algae separately, like that. And we will use the CTAB method, or there is any kit for that.
See, for DNA extraction from lichens, no need of culturing it. If you culture the lichen and the microbiome part, then it is good. You will get a pure DNA. There is no problem. But using the lichen itself, you can extract the DNA. You can extract the DNA both by kit methods as well as by the CTAB method. And see, only thing is that you have to take care in case of lichens. First thing is that you have to surface sterilize it very nicely so that there should not be any contamination. Second thing is that your lichens has these secondary metabolites, lot of secondary metabolites, which actually hinder the quality of the DNA. You have to remove it by dipping it for a few minutes in the acetone, and then the CETA method or the kit method you can use. In the if you have a material in good quantity, CETA method is very good because you will have the good quantity of DNA. But if you don't have good quantity of materials, raw material or the lichen thallus, then CETA method is good. And I mean, this one kit method is good. So there are no special kit for lichens. You can use the fungal kit only. And once you have DNA, that is total DNA, it has both this fungal part as well as the algal part. And this is the primers which are very much important to this amplify the fungal genes. So that is it. Okay.
Sir, and sir, one more thing, that is means for learning the molecular studies on lichen, is there any workshop or skill development program through CSIR or any other organization?
See, we will be coming up with such kind of training programs in future very shortly. Right now, as such, there is no training programs, but you can contact ARI, Dr. Rajesh. You can contact him. They may help you in case of if you need. They already started doing this kind of research.
Okay, sir. Thank you very much, sir.
Thank you. Dr. Rajesh Kumar, if you're around, before I let you ask the question, please let us know whether you're going to conduct some training programs on lichens at ARI soon.
Hello, sir. Sanjeeva sir, thank you very much
Yeah.
for a wonderful
Yeah.
talk. Thank you, Dan, for the question. Yes, sir. We intend to conduct training sessions very soon. Initially, we will start with the fungus, and in the November we will have hopefully we are in discussion with the Sanjeeva Nayaka sir, because we had a wonderful session at Kerala Forest Research Institute. Therefrom, we discussed about the possibility of exploring this area, and hopefully we will have a discussion with Sanjeeva sir, NBRI team and ARI. Together we will plan something very soon, sir. Hopefully in the last quarter of this year, hopefully.
And what is your question to Dr. Sanjeeva Nayaka?
Yeah, Sanjeeva sir, my question is, because we have extensively working on crustose lichens of Western Ghats, and we have also explored the mycobind and photobind association in different genera. So I just want to understand whether we can correlate these kind of data with the data available from the Indian Himalayan regions, because when we get such a data, we can even predict some kind of evolution which happened within India, as well as we can connect it with the continental drift and this biotic furry concepts which is existing in lichens of India. So what is your comments on these areas, sir?
Yeah, certainly. See, we need not wait till complete entire country to be explored for the lichen and make a inventory. So whatever the data so far we have, that can be utilized for comparing the uh, diversity in different places, and also using some strong statistical softwares. Certainly, this can be done. So, for example, Dr. Luking at Germany, he has done such kind of studies already. Following that, or improving that, we can do it for India. And one thing is that we can compare within India. Second thing is that we can compare with our neighboring countries, and then the whole world. So certainly, this is a good idea. One can do it using the good statistical applications.
Sir, do you have photoband and microband data for Indian Himalayan areas for many of the crustose lichen species? I mean, I'm talking in terms of species level identity of algae and mycobiome.
No. So what we know so far is that only that can be extracted by looking into the lichen species. For example, you take any lichen, crustose or foliose, mycobiont. You know the name of the lichen is itself the name of the mycobiont.
Yes, sir.
Photobiont, we know the genus name, whether it is Triboxia, Cytonema, Nostoc, or whatever genus name is there, but species name is not there.
Yeah. Sir, actually, we have faced a problem when we sequenced plenty of the photobiont. Most of them are obviously in Graphidaceae trentifolia. So when we made a tree, we understood that there is another genus which is called Princina, which is coming as a like paraphyletic clad within the trentifolia group. So do you have any idea about this kind of relatedness?
No, I don't have, but what I can suggest to you is that since I also have a same project nowadays,
Okay.
recently started. See, now we have to recognize our photobiont at a species level. So
Yes,
that is so the whatever the marker or the primers whatever we are using, that is not sufficient to differentiate photobiont at species level.
yes, sir.
We have to modify our research. We have to find out some markers which can differentiate the at a species level. So then only we can do it. Otherwise, all Graphidaceae, all lichens you will get, all Graphidaceae will tentrifolia, all Parmeliad lichen will get Triboxia. So that is not helpful. So we have to identify at a strain level now. Lower
Yes,
than,
sir.
yeah.
Yeah, in fact, ITS sequences which we have got from many of the Graphidaceae photobind was very. I mean, it was resolving up to species level in some of the clades, not in all the clades. That you are right, but I wonder that even in international level there are any additional primers available so far.
It may be there. You have to look into literature. It may be there. I doubt. I mean, I'm sure it should be there.
Okay.
If
Thank
not,
you, sir.
if not for phytobiont or for photobiont, you can see other literature, other organism, and then other method also, which is applied for the other group of organism, higher plants. We can adopt that, modify that, and then you can see that.
Yes, sir. We are extensively doing it for fungi as secondary barcodes. We are sequencing and we use this super barcode concatenated data for resolving the phylogeny. Uh, Obviously, for Indian lichens, I think we should collectively do it. Thank you very much, Nayaka sir, for giving this presentation. And I'm sure that our KFRA discussion were instrumental in coming up with this talk. I have had a detailed discussion with Damodar sir, and he was very supportive in conducting such session. Thank you, Damodar sir, for being generous with us. Thank you.
Thank you, Dr. Rajesh, managing editor of MicroAsia, for this wonderful discussion. Your contribution to this discussion going on. Ladies and gentlemen, we have five more minutes for the discussion. Okay, so I can see 4 people with raised hands. Please limit to 1 question, and Dr. Sanjeeva Nayaka will be happy to answer. Mr. Vinayaka KS, please ask your question.
Good morning, sir. Thank you for nice presentation, sir. Sanjay Naik, sir, very nice, informative talk. Just my question is that you go through your presentation. I got so many information, but I did not get information about the aquatic lichens. Especially, sir already told that marine lichens. Similarly, is there any aquatic lichens is reported from India, sir?
As I told you, yes, very few species are there. Very Verucaria species are there. 1 or 2 species are there, not more than that. Especially in Verucaria, we have it, and we are coming with a paper, and then only I'll be able to exactly tell you. But there are 1 or 2 species are there of Verucaria, not more than that.
Yes, sir. Yes. Sir, is there any recent work on these aquatic fungi in India, aquatic lichens in India, sir?
See, once we tried, we were not successful. And uh, so, I mean, there are no research happening like that in India. So you can take up, that is a very interesting area. You can do it. You can take up. No. Okay.
Yes, sir. Thank you, sir. Thank you.
Thank you. Dr. Sudipta Sinha, please ask your question.
Thank you, sir. Thank you, sir, for such an insightful presentation. And as for limited time, sir has requested for one question. I'm just asking a very basic question. Sir, is there any possibility that we can apply for women scientist project on lichen identification? And I'm from a small part that is from northeast Tripura. We are having like very few infrastructure or labs working over it, so can we do it in collaboration with some other institutes? And also I think Dr. Rajesh S sir has mentioned they are going to organize some training also, maybe we will be helpful with it. So is there any possibility we can do in collaboration?
Yes, certainly it's possible. So if you're interested in applying for a women scientist project, you can contact me and I will guide you how to write. See, simply you write identification project, you will not get. You have to incorporate few other aspects to make it interesting. Second thing is that collaboration possible. Certainly, we will help you in identification of the lichens and all. Women scientist project cannot be collaborative project because it has to be done there itself, but you can show that you will be taking a help from CSIR-NBRI, so that is sufficient. So you certainly you can do it. You contact me, and I'll help you in this matter. Okay.
Thank you, sir. Thank you so much.
Thank you. Madam Shashireka, please ask your question. Only one question here.
Yes, only one question. Thank you so much, Dr. Nayaka. It was a very interesting talk. I just wanted to clarify my doubt. Like whenever I go on the field, like I go to Keshav Srishti where they have a betel nut plantation, and there are these lichenic patches on that. The common question that I'm encountered with is whether it is parasitic. Are there any parasitic lichens, or could lichen be also parasitic?
See, no, lichens cannot be parasitic because, but dense growth of lichens may cause some problem, but lichens are not parasitic. In the betel nut tree plantation, what you see, the lichen, they're all superficial growth. So, see, there are clusters like in white patches, what you see more. But
Yeah.
what happens, why lichens are not parasitic? Because they don't draw the nutrition from the host plant on which they are growing. They use the host plant only for anchoring, attachment purpose. So they don't have that root kind of structures which penetrates deep into the bark of the tissue of the bark and absorb the nutrition. So, these when the lichens densely grow on the tree bark, we can switch off your mic. So what I mean to say that the lichen cannot be a parasitic because they don't depend on the tree. Only once a dense growth of lichen is there, it may sometimes block the lenticels of the trees and also otherwise no problem. There is no reports of parasitic lichens.
Okay. Thank you so much. Thank you so much.
Ladies and gentlemen, the last question from Biplab Mahato, if you're around. You have been waiting for long. Sorry.
Hello, sir. Thank you for the session. So I have a quick question. Do lichens are bioluminescent? Do they glow under UV light?
Yes, some of the lichen, they glow under UV light, but they're not bioluminescence. You're mixing two words here, so two aspects here. See, bioluminescence is something different, where see when during the daytime they take up the sunlight and then the nighttime they glow. So it is not case of lichens because these kind of questions I get all the time. So the bioluminescence mushrooms are there, fungus are there, fungi are there, but not lichens. But lichens, there are several species of the lichens. They emit fluorescence when you see them under the UV light. So it can glow yellowish or sometimes pinkish, sometimes orange. So that is there. That is possible.
So, and you mentioned about a lot of recipes which they uses in Indian context. They uses lichens. Do we have any local names, or do they refer to any local lichens in Indian context?
Yes, yes. See, it's all depends on your language, what you use. But the common language, what they use for the lichen is that stone flower. You right now, you Google it, or you search in the Amazon or the Flipkart, stone flower, you will get that spice, the lichen spice. So even in the supermarket you go, whether in Lucknow we have Lulu Market, I've seen the drums of lichens they're selling as a spice. You go to Big Bazaar or a Spencer, you will get a packet of lichen as a spice. They sell it. Stone flower it is usually called. Otherwise it can be called as dagat pole. It can be called as um, chadila, julagas, or many other things. But stone flower is a common name. You will get it.
Thank you, sir. Thank you so much.
Ladies and gentlemen, I'm sorry that I must stop this discussion. I mean, it has been going on so well and we are learning so many new things, but for the sake of, I mean, as the time is not there with us today for this session to be continued, I have shared the web page of Dr. Sanjeeva Nayaka, NBRI web page. So please visit that web page. You can find his email address there. If you have, I mean, I see many questions in the chat box. I'm sorry, ladies and gentlemen, we will not be able to take up those questions right now. Please contact Sanjeeva Nayaka by email. I'm sure he'll be happy to answer some of your questions there. And
Sure.
yeah, maybe you can find him in ARI in coming months, or you can find him in NBRIs when he conducts some of his programs in coming years. Dr. Sanjeeva Nayaka, thank you very much. So much. We have been kind of blessed to hear your experiences, the kind of information you have shared with us. We are richer in knowledge because of your presence and your presentation today. Thank you very much for your kind presentation.
Thank you, Dr. Belle, Damodar Shenoy, and Rajesh, and others of the MicroAsia for this opportunity. Thank you, all the audience, for the patience hearing. Thank you.
Yeah, I wanted to just express my immense gratitude to the audience, today's audience. I expected only 30 people, but we have been getting more than 50 people in the audience. So that's a kind of a great relief for me. And it is good to have a good number of people in the audience, especially asking some insightful questions and provoking questions to the speaker. That way we can learn together. Thank you, ladies and gentlemen in the audience, for your kind participation today. And we will be having a series of lectures in coming weekends, actually. So please try to participate and enrich the knowledge. I mean, we'll learn together. Thank you very much. With this, we sign off. Dr. Sanjeeva Nayaka, have a nice Sunday there at Lucknow. Yeah, bye-bye. Namaste. Yeah. And ladies and gentlemen, with this, we sign off. Thank you very much. Bye-bye.